Hey Quinn,
Hi,
how are you?
I'm good. How are you?
Good. How'd your interview go?
It was really good. And actually, I have another one tomorrow. And I don't know if it'll be over by the time lab starts. It might be,
Okay.
but I'm not sure. So I might not be there tomorrow, but I also could be there.
Well, all we're doing is just studying taxonomy and identification.
Okay.
So it's just
Okay.
going to be like most of the lab was last time. You're going to be sitting there, you know, getting to know the amphibians, and so it's not really super structured. I'll be surprised if everybody stays the whole time.
Okay.
Lab is going to be open from 7:45 until whenever the last person leaves, and
Okay.
one of the other things is you can go in and make up anytime you need, anytime you want. So,
Okay,
um,
cool.
my advice generally to people is, and I told you guys this in lab last time, is to break those sessions where you're learning identification up into shorter periods of time um, more frequently rather than saying, oh, I'm going to spend 2.5 hours on it today and 2.5 hours on it next week. It's one of those situations where you get it socked into long-term memory much better by visiting it more frequently and for shorter periods of time. So, so for example, tomorrow I'm going to be there at not 7:45. What am I saying? 7:45. 10:45. I'm going to be there at 10:45, and you're free to come to class. Quite frankly, anytime during that span from 10:45 to 1:35. I don't actually expect you to be there the whole time because it's tiring, it's fumey, and spending an extra hour on it in that one session doesn't necessarily gain you as much as doing a shorter time period on a fewer number of specimens, really concentrating on those, then going away for a couple of days, and then maybe coming back and doing some time in lab on Friday. So um, that's been my experience having taken a lot of taxonomically based courses is that biting it up into smaller chunks more frequently generally gets you further than a fewer number of long sessions. Um, when I took ichthyology, which is the study of fishes, as an undergraduate, me and a guy named Paul Sill, we would spend half of each lab period sitting in lab working on our identification skills, and we would always bug out about halfway through class and go to the local stream that flowed through the campus where we were students, and we would go fishing for the remainder of the period. And I would gather that we learned as much about fish during those fishing outings as we did sitting in lab learning identification. And in terms of identifying things, so um, the ichthyology professor, when he got to the section on fisheries, towards the end of the class where he talks about kind of commercial fisheries and state departments of wildlife and things like this and stocking of game fishes and lakes and things like this. In Texas, there's nothing but reservoirs. We don't really have a lot of big rivers, and we don't have any natural lakes really. The one natural lake that people talk about is actually a natural lake that was formed from a log dam that was from people transporting logs. So that's not even a natural lake. It was a lake that was formed as a result of a human accident. But anyway, so all of the big bodies of water in Texas are made up of reservoirs, dammed rivers, and so he had watched me and Paul work in ichthyology lab all semester and was aware that we always bugged out early and went fishing at the stream that ran through our campus. And so when he gets to the fishery section, he's like, um, so when you talk about fisheries in Texas, you're really talking about large reservoirs because Paul and Paul are the only people who do small stream fishing in Texas. And so anyway, um, so know that tomorrow I will not be judging you if you show up late, and I will not be judging you if you leave early. Everybody does learning identification differently. Do whatever schedule works for you. Just know that in 3 more weeks, you're going to have a lab quiz where you're going to be asked to identify and tell me information about the specimens that are in the lab. Questions about that? All right, questions about taxonomy and systematics from last time? Can you guys hear me?
Yeah.
Okay, all right. I just don't have any feedback. Okay, so I see faces, but like, okay, no questions. Good. We'll continue. Let me share my screen with you. You're going to have to do a little bit of work today. So you might want to have a piece of paper or some writing implement, whether it's your iPad and an Apple Pencil or whatever, because we're actually going to work through a problem of sorts. So you guys will recall that last time I told you that we want to generally be using taxonomic systems that are natural taxonomic systems, systems where the taxonomy of the organisms reflects what we understand of the evolutionary history of the organisms. And so the way we establish this evolutionary history in the form of phylogeny is a pretty regimented system for figuring out what happened in the past based on what we observe in organisms that are extant, that are present today. So when you construct a phylogeny, you basically look at different characters and you compare these characters to an outgroup. And so if we're dealing, for example, with the vertebrates, those organisms in the phylum Chordata who have a vertebral column, we are going to generally be using as our outgroup something like the non-vertebrate animals, other chordates, or if we're dealing, chordates. And so the lamprey, the sea lamprey, is a chordate, but it has a notochord, which is one of the characteristics of the chordates, but it doesn't have a vertebral column. It also doesn't have jaws. It also doesn't have a bony or a cartilaginous skeleton. So sea lamprey is this, it's a type of fish that sucks onto the side of other fishes and feeds on their fluids, essentially. And so that might be the outgroup of all of the vertebrates. So the vertebrates are going to include all of the fishes, the amphibians, the reptiles, the birds, and the mammals. And so you always want to identify the outgroup because the outgroup presumably has the ancestral form of whatever characters you're going to be looking at in the in-group. Now, members of the in-group might share some of those ancestral characters with the outgroup, but you use the outgroup to polarize your characters so that you can say, well, this character evolved from this into this. And so that's the whole purpose of having an outgroup is if these are jawless fishes, then the presence of jaws is a character that evolved that sea lampreys and other jawless fishes did not have that all of the vertebrates then do have. And so the polarization of a character is the directionality of the character, a character changing from this state to this other state. And so we group organisms by their shared derived characters. These derived characters are those characters, or those states of the characters that aren't the ancestral character, that aren't the character state that was found in the outgroup. And what we do is we begin with the most inclusive of these shared derived characters, and we group organisms together on the basis of which groups share a particular derived character. And we decide that we have the best phylogeny by calculating up the number of evolutionary transitions, and whichever tree topology, whichever shape of the tree has the fewest number of evolutionary transitions is generally viewed as the best tree topology. And this is referred to as the parsimony criterion. Parsimony is just the idea that the simplest explanation is oftentimes going to be the correct explanation. And in a phylogeny, the simplest explanation is the tree topology, the shape of the tree that has the fewest number of evolutionary transitions, character state changes, or steps. In the literature, you'll oftentimes see people refer to the number of steps in a phylogeny. Questions about this? These are kind of the overarching principles of how to build a phylogeny. Does this sound at all familiar? Okay, so you've heard this before in a class. Okay, so the first thing that we're going to do is we're going to build a character. So we're gonna, this particular example starts with amphibians and moves through mammals, birds, lizards, snakes, turtles, and alligators. And so what you generally do is you begin with a set of characteristics, and then for each of these taxa, you ask yourself, well, what is the state for this character in all of these diverse groups of taxa? And so, for example, where does the skull joint occur, and is there one? How many digits are on the hind limb? Is there a gizzard present? Is there a bladder present? What type of waste do they produce? Do they produce ammonia? Do they produce urea? Do they produce uric acid? What type of metabolism do they have? Are they ectothermic, or are they homeothermic or endothermic? What type of egg do they have? Do they have a hemipenis? Is the quadrate bone fused or not? How many temporal holes are in the skull? And so these are the kinds of morphological characteristics that have been used to build simple phylogenies of all of these different groups of organisms. And I could send you to a textbook, and you can find these kinds of characteristics in like an anatomy textbook, which I think all my, oh yeah. So we could go to the analysis of vertebrate structure, and we could look up each of these characteristics, and we could see which groups actually possess these. This is a comparative anatomy book, and it's actually designed to compare the anatomy among different groups of vertebrates. But we won't do that because that's time consuming. It's kind of a pain. So I'll just give you what the matrix is. So in the amphibians and mammals, there are 2 skull joints, and everybody else, there's only 1 skull joint. The hind digits vary in number. Snakes don't have any hind digits because they don't have any legs. So they don't have 4 digits either. So amphibians have 5 digits on their hind feet, mammals have 5, lizards have 5, turtles have 5, alligators have 4, birds have 4, and snakes have none. 2 groups have gizzards, birds, and alligators. Bladder is present in everybody except, once again, birds and alligators. The type of waste that they produce in terms of kidney function, some groups produce urea, some groups produce uric acid. Are you ectothermic or are you endothermic? Do you produce a shelled egg? So this is a shelled egg as opposed to an unshelled egg. An egg with a hard calcareous shell, or a parchment shelled egg versus a naked egg. What's so funny, Rose? Nothing. Okay. Hemipenis. A hemipenis is a structure where the copulatory structure of the males is divided into 2 halves. We'll see some pictures of hemipenises when we get to reproduction in the reptiles. They're crazy looking things. The fusion of the quadrate bone on the skull, is it fused or not? And then the number of what we call temporal fenestrae in the skull, these openings in the skull. Do you have none? Do you have 1? Or do you have 2? And so these are the characters that we have. And so just to simplify things, what we generally do is rather than have these characters be 2 versus 1 and 5 versus 4 versus 0 and no versus yes, we generally code these as zeros and ones, where 0 refers to the ancestral condition and 1 refers to the derived condition, the evolutionary derived condition. And we do this by comparing it to the out group. And in this case, the amphibians are the out group. And so there are multi-state characters like the hind digits where we would then, so there's 0, 4, and 5. 5 would be the ancestral condition. And then 0 and 4 are derived. And so 5 would get a value of 0, and then 0 and 4 would get like 1 and 2, just to distinguish them. And so we would transform this matrix of characters into a set of zeros and ones and twos. And so this is just the same recoded, the same characters that we had before, except they're recoded. So for example, if you look at character 10, 0, 1, 2, 2, 2, 0, 2 corresponds to 0, 1, 2, 2, 0, 2, because that's the number of temporal holes. That was a terrible example. Let's look at the hind digits. 0, 0, 1, 0, 2, 0, 1 corresponds to 0, 0, 4 is 1, and no hind limbs is 2 in this recoding. In reality, if we don't know whether evolution went from 5 fingers to 0 fingers or went from 5 fingers to 4 fingers to 0 fingers, since we don't know what that relationship is, we can't really say that 1 happened before 2 happened. All we can really say is that we know what the ancestral condition is, and we know that there are 2 derived conditions, but we don't know the polarity between those 2 derived conditions. And as it turns out, because 0 digits only occurs in snakes, that's actually an autapomorphy. Remember this term from last time. This is an autapomorphy. And remember that an autapomorphy is a derived character that only occurs in one taxon. And as a result, because it occurs only in that one taxon, it's not really useful for grouping because it only occurs in one group. And so we would actually just ignore that one autapomorphy and just pay attention to the other synapomorphies that are useful for grouping, for separating ancestral groups and more evolutionarily derived groups. So this is what this whole thing looks like. And one of the nice things about converting it to zeros and ones is that computers are really good at dealing with zeros and ones. And once you get up to a certain number of characteristics, it isn't really useful to do this by hand. It becomes too complicated. But the other nice thing about converting it to zeros, ones, and twos is that it's easier to see patterns. And so what we generally want to do is, in this case, we're going to start with the amphibians, which is the out group in this particular analysis. And the first thing that we have to do is we have to say, what are the characters that separate the amphibians from everybody else? What would be the character or characters that we would use for this?
The egg.
The egg. So that is character 7.
Yeah.
Character 7 is the egg. And in this case, character 7 is transitioning from the state of 0 to the state of 1, which is going from an unshelled egg to a shelled egg. And so this is the difference between the amphibians and what is broadly known as the amniota. All of the other groups that are on this table have amniotic eggs, meaning an egg with the presence of a chorion and an amnion. Amphibians have an egg that only possesses a chorion. So if you've taken, I don't know, how many of you have taken developmental biology with Dr. Vora? Did he talk about differences in egg structure?
Yeah, a little bit.
Yeah. So this is a huge difference. It allowed these animals that are in this group to divorce themselves from water, because the amnion is an extra embryonic membrane that basically provides a buffer between the embryo and the outside environment. So it's kind of the difference between being a fully terrestrial organism rather than an organism whose reproduction is still tied to water. So now that we've separated the amphibians from amniotes, what characters next do the next biggest grouping appear to be? If you look across the matrix of characters, what are other characters that tend to group a lot of the taxa together?
One and five.
Yeah, 1 and 5. So 1 is the skull joint and 5 is the waist type. So who does it separate out?
Amphibians and mammals.
Pardon?
Amphibians and mammals.
Okay, so it separates out mammals, and it separates them from everybody else. And it separates them on the basis of character 1 and character 5. Both of these are going from a state of 0 to 1. And for character 1, that is the skull joint, which goes from 2 skull joints to 1 skull joint. And for 5, which is waist type, it's going from urea to uric acid. So that's just what these 0 to 1 transitions mean. But typically how you would see this in a tree is you would see the first one of these was character 7 going from 0 to 1, character 1 going from 0 to 1, and character 5 going from 0 to 1. And these hashes on the evolutionary branch here, these hashes refer to evolutionary transitions or steps. So I'm going to actually just redraw this. Now it looks messy. So now in the groups that remain, birds, lizards, snakes, turtles, and alligators, what characteristics are going to be useful for grouping within those groups? So looking from here down, from here down, what characteristics serve as good grouping characters? So 10. So one of the things that happens with 10 is that it puts everybody together except the turtles, doesn't it? So everybody has 2s except for turtles, which have 0. So we can map 10 on here, and 10 separates turtles from everybody else. So turtles have, well, what is 10? Oh, so turtles have 0 temporal fenestrae. Turtles have 0 temporal fenestrae. And everybody else in that group has 2 fenestrae. So this is going from 0 to 2. But 10 also appears someplace else, but 10 is also an odd apomorphy. And so in mammals, we have only 1 temporal fenestra. And so in the mammals, 10 goes from 0 to, this goes from 0 to 2, goes from 0 to 1. So in mammals, we developed this temporal fenestra, which the ancestral condition is presumably 0 because that's also what amphibians have. But then there's also another independent transition here going from 0 to 2 that separates turtles from all of the other groups of terrestrial vertebrates, all the other amniotes. So now we've taken 10. So what I would typically do if I was doing this on paper is as I mapped each of these onto the phylogeny, I would draw lines through them. So now among those groups that are left, when you take turtles out, what would be a good grouping variable that would separate birds, lizards, snakes, and alligators?
6.
Okay, but in that group, let's see. In that group, though, if you just look at birds, lizards, snakes, and alligators, that appears kind of as an odd apomorphy. It only occurs in birds. So once again, that's not useful for grouping.
So would 3 and 4 work?
3 and 4 would work. What else would work?
Eight and 9.
3 and 4 and 8 and 9, but recognize that they're going in different directions. So what do 3 and 4 and 8 and 9 group or separate?
Eight and 9 is lizards and snakes.
Actually, both 3 and 4 and 8 and 9 separate lizards and snakes from alligators and birds. So what happens with birds and crocs is for birds and crocs, we look at characters 3 and 4, which go from 0 to 1. And then characters 8 and 9 support this grouping of lizards and snakes together, where once again, 8 goes from 0 to 1, and 9 goes from 0 to 1. Okay. Now, are there any differences between alligators and birds?
Number 6.
Okay, so number 6. And in number 6, we have birds that are homeothermic or endothermic. So we have character 6 going from 0 to 1, but where else do we have character 6 appearing? This transition from ectothermy to endothermy for character 6.
Mammals.
In mammals, we also have 6 going from 0 to 1, okay? So at this point, we've accounted for character 1, character 2. No, not character 2. Nope, no character 2 yet. Character 1, character 3, character 4, character 5, character 6, 7, 8, 9, and 10. Oh, so the only one that we haven't accounted for is character number 2, which is the number of hind digits. And in this case, we see that mammals have 0, and no, it's just complicated, isn't it? Lizards have 0, snakes have 2, which is 0 actually. So let's see. So once we've mapped all of the other characters on, we just have to fit in whatever's kind of left that has unusual sorts of things. So amphibians, mammals, turtles, and lizards all have 0. So you don't have a transition to 1 until you get down to here. So right here would also be character 2 going from 0 to 1. But then we also have snakes and lizards being divided at this point because lizards still have the ancestral characteristic, and then snakes have character 2 going from 0 to 2, which is basically the idea that snakes are limbless. Or no limbs, and as a result, they have no digits on their feet. So now we've accounted for character 2 on the tree. And once again, this thing, now that it's finished, it speaks to this idea that I had before about, why do we study herps and why are herps kind of circumscribed the way that they are? One of the reasons is that back before we developed phylogenetic systematics, people oftentimes group birds and mammals as being more similar to one another just on the basis of one characteristic, the fact that they were both endothermic. Birds are endothermic, mammals are endothermic. But that ignores all this other aspect of their biology, their morphology, their physiology, that point to the fact that they're actually closer related to one another in evolutionary terms to the crocodiles and alligators than they are to mammals. All of these groups, turtles, lizards, snakes, crocodiles, and birds, for example, all have red blood cells that have a nucleus. So we could have put that as one of the characters up here. In mammals, the nucleus is extruded from the red blood cells, and our red blood cells don't have nuclei, and as a result, they have to be constantly replenished. So if we count up the number of steps in this phylogeny, there's 1 step here, 2, 3 steps here, 4, 5, 6, 7, 8, 9, 10, 11, 12. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So this tree has 13 steps. Now, if we took a different approach to this, where we actually moved birds to the point that birds were the sister taxon to the mammals, one of the things that we would be able to do is we would be able to get rid of character 6 down here, which is metabolism. Character 6 appears here and here. So we could get rid of one of these 6s because birds aren't down here. Crocs are now down here by themselves, crocs and alligators. But in the process of that, we would have to now say, well, birds and mammals both evolved homeothermy, so we would then have to add 6 here, going from 1 to 0, reversing itself. It's what we call a reversal, when a trait, wait, is that right? No, we don't have to leave that at all because that only occurs on that one branch. So that doesn't cause any problems. But what we would then have to have happening is on the bird branch here, we would have to have the transition of 3 from 0 to 1, 4 to 2, and 0 to 1. All of those transitions would have to happen on the bird branch here, and they would also have to happen on the crocodile branch here. And so by moving birds up there, we would now have a tree that was 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. This would now be 15 steps. So by moving endothermy, supporting a a monophyly of birds and mammals together that reduced one step due to character 6, but it added 3 more steps, giving you a net gain of 2 steps. So what we would say is this is not the most parsimonious tree. The most parsimonious tree has endothermy evolving in 2 places independently, a convergent characteristic. Down here, separating birds from crocs. But as a result, we don't have to move these 3 characters to another part of the tree in order to support a monophyletic grouping of birds and mammals. So this is what the concept of parsimony is and how it's used in phylogenetic reconstruction. The phylogeny with the fewest number of steps is the phylogeny that we adopt. Does that process make intuitive sense to you? Okay. So let's go back to the concepts. When we construct a phylogeny, we always compare character states to some outgroup. In this case, the amphibians were the outgroup. We only group organisms by their shared derived characters, apomorphies, and more important of these apomorphies are synapomorphies, which are the shared derived characters. And actually that should say synapomorphies because I have shared and derived there. And we begin with the most inclusive synapomorphies, and we ignore odd apomorphies because they only occur in one taxon. And then the criterion for our accepting a particular shape of the tree is based on the shortest number of evolutionary transitions that are necessary to get that tree to work, essentially. And we've applied all of those concepts to this data set, which we recoded into this data set, which gave us this phylogeny. And this is the phylogeny that most people who teach vertebrate biology adhere to. Mammals actually were around during the time of the rise of reptiles. They were already on the landscape. They just were small and inconsequential because other groups of organisms dominated the Earth back then. And so mammals go back quite early in evolutionary history with all of these other groups evolving after mammals appear. They just don't come to be the dominant form of life on the planet until much later in geological history. And we get away from kind of this view of using similarity, subjective similarity, to group organisms because we no longer group birds and mammals together simply because they're homeothermic or endothermic like we are. Endothermy turns out to be a physiological state that has evolved independently at 2 different times. And so this is what we refer to as a homoplasy. A homoplasy is just a characteristic that evolved convergently in different groups. And the process of building phylogenies this way allows us to identify when convergent evolution occurs. Why do certain organisms have similar structures, but they didn't get those structures through common ancestry? They didn't inherit them from a common ancestor. They actually evolved them differently. So for example, what are 2 groups of flying organisms that you can think of that are not evolutionarily closely related, but have similar biologies because they can fly?
Birds and flying squirrels.
Okay, so flying squirrels don't actually actively fly, they just glide.
They glide.
They glide. But so birds is one group that can fly. What's another group?
Insects.
Okay, so insects. What's another group? Group of mammals.
Bats.
Bats. Okay. So all of these organisms fly. And if we used flight as one of the characters in a phylogeny, we might want to group these together. But then when we look at the anatomy in detail of how these organisms have a flying apparatus, when we look at the structure of the wing, we see that the insect's wing is basically made out of the exoskeleton of the organism. The bat's, the wing, if you look at Batman, if you guys are familiar with the Batman logo, the shape of that bat wing, let's go find a bat picture. Oh, come on now. What you'll see with the bat wing is that this is a made-up bat wing, but basically you've got the humerus, you have the fused radius and ulna, but the main bulk of the wing, those are basically just really long fingers spread out. These are actually elongated phalanges. And so the humerus and the radius and ulna fused are making up the base of the wing, but the main area, flight area of the wing is made up of elongated phalanges. You see that long phalange there, then a shorter phalange there, and then a short phalange there. So it's basically the outstretched hand that makes up the wing of a bat, as opposed to a bird wing is essentially made up of the bulk of the wing is made up of the radius and ulna, and the bones of the hand are actually reduced and fused. So your metacarpal bones, these long bones on the back of your hand, are all fused together, and you only have one finger bone at the very tip of the wing. And so the structure of the bat wing and the bird wing, even though they're made up of the same bones, the importance of those bones and the structure of those two types of wings is very different. So even though these are all made up of arm and hand bones, the bird wing is made mainly up of the arm bones, and the bat wing is made up mainly of the hand bones. And so even though they all have similarities in their biology, similarities in their structure, et cetera, when you begin to look at the detail of those things, you find that they're very different. And it's because this characteristic flight has evolved a number of different times. And within the vertebrates, it's evolved twice in bats and birds. But because those are convergent characteristics, they both have wings. Because they're convergent characteristics, they evolved independently of one another. It turns out that the structure of those wings is very different. And that's also the thing that you oftentimes see in convergences or homoplasies. Questions about building phylogenies, how it's done, what to look for, what the procedure is. Okay. So when we think about natural taxonomy, because, for example, these lizards are all in a monophyletic group, it's hard to justify making these two groups into these two taxa one species and the rest of these taxa another species. But sometimes that is done. But it is much easier to think of this in terms of natural taxonomy when one group of specimens or one group of taxa forms one monophyletic group, and the other taxa form a different monophyletic group. Remember that a monophyletic group includes a common ancestor and all of the descendant taxa. So this is a monophyletic group because it includes the common ancestor and both descendant taxa. This is a monophyletic group. It includes both descendant taxa and the common ancestor. But this group of green lizards over here in this phylogeny is not a monophyletic group because it includes the common ancestor of all of them, but it doesn't include all of the descendant taxa. And so when we think about natural taxonomy, we would say that separating these two things into species 1 and species 2 would not be a good natural taxonomic grouping because it includes some, but not all of the descendant taxa of a common ancestor. These would both be valid species because both of these taxa, species 1 and species 2, includes the common ancestor and all of the descendant taxa, and the common ancestor and all of the descendant taxa. And that's what natural taxonomy would want to do, erecting a taxonomy that reflects monophyletic groups rather than paraphyletic or polyphyletic groups. So one of the important things that happened in vertebrate evolution was this evolutionary transition from water to land. And this was a pretty big transition because it involved lots of things about the morphology and a lot of things about the physiology of organisms. So it was a big event in vertebrate evolution. When you move from the water to the land, your body is no longer supported by the water, so you need much stronger skeletons. You need pelvic girdles and pectoral girdles. And if you're going so you need them to begin with because they support the limbs. And once you have limbs, you need those pectoral and pelvic girdles to be bigger and stronger, once again, to support the weight that is being supported by the limbs. And then obviously, you're facilitated in a terrestrial environment through the evolution of limbs. This is especially when you consider comparing these to the organisms that dominated the earth before terrestrial organisms came into being, which was the fishes, which are limbless. They have fins instead of limbs. Then there are also all sorts of things that happen with terrestrial feeding. And so the shape and structure and bony apparatus of the skull comes into play. So you see a lot of change in cranial morphology, jaw morphology to facilitate feeding. And then there are, of course, evolutionary changes in the structure of the egg to avoid desiccation during reproduction. And there are also alterations to the skin to avoid desiccation. So you're made up mostly of water in terms of your wet weight is mostly water. And you have to maintain water balance. And so if you're on land, it's harder to maintain that water balance than if you're in an aquatic environment. And so both in terms of reproduction, alterations to the egg, and in just everyday life, which involve alterations to skin structure, those things had to occur as well in order for organisms to transition from an aquatic environment to a terrestrial environment. And so what we see here is the fishes. So we have the earliest precursors of vertebrates, which are the coelacanths. You guys know what a coelacanth is? Anybody? Coelacanths. Go look up what a coelacanth is. These are cool fishes. They were first discovered. They were new to science back in the 1970s. Although we knew about them from fossil evidence, but the first live ones were found back in the 1970s. These are the lobe-finned fishes. And so they're these weird funky fishes. They have a broad lobe that supports the fin. And when you look at the structure of this lobe, it actually has a bony structure that is similar to the arm of the terrestrial vertebrates. So these are presumably the evolutionary precursors to the limbed organisms that actually came out onto land. Like I said, we had records of these from fossil beds, but the first living organisms weren't found until the 1970s, where some deep sea fishermen running dredges, these fishing nets that drag along the bottom surface of the ocean pulled one of these up, and there have been, I don't know, 50 or so found since then. If we go to the KU collections and we happen to be shown around by a ichthyologist, which is what happened last time I went there, we were interested in going to the herp collections, but once he was finished taking us through the herp collections, he was like, you know, I'm an ichthyologist, so I can't really speak a lot about the herp collections, but I'm an ichthyologist. Do you guys want to see my favorite fish in the collection? We're like, yeah, sure, why not? And so he took us over into the fish collection, and he pulled out their collection, their specimen of a coelacanth, and it's in a big metal vat. And he pulled it out and let the students look at a coelacanth. They're pretty cool fishes from an evolutionary standpoint. So the coelacanths are these lobefin fishes, the dipnoi are the lungfishes, and all of these organisms throughout all of this, these are all the terrestrial organisms, are all aquatic organisms until you get to about right here, in which case organisms start crawling out onto land. And the first group to do this was, of course, the amphibians. And even though they can exist terrestrially as adult forms, they don't reproduce on land, generally speaking. Or if they do reproduce on land, it's always in pockets of water on land or moist environments that the animals themselves create on land. And it isn't until you have the evolution of the amniotic egg that you begin to get organisms that are capable of both reproducing and living on land. And so this particular evolutionary event, the development of the amnion, kind of marks the movement of organisms leaving aquatic environments completely and living their entire lives out on land. Even though you have salamanders and frogs that have thick enough skin that they can avoid desiccation, you don't have them escaping water for reproduction until you get the development of the amniotic egg. And at that point, you get the development of dinosaurs and you get the development of reptiles, which includes lizards, tuatara, lizard snakes, tuatara, birds, and the other reptile groups. So when we go back and look at the development of things, we see that we have the early fishes that basically had fins without any sort of bony underpinnings. And then we see that there's the development, once again, in the Devonian, this is hundreds of millions of years ago, we begin to start seeing these bony components underlying the fin structure of both the pectoral fins and the pelvic fins. In this case, it's the pectoral fin. In this case, it's both the pectoral and the pelvic fins. And they've color-coded for you what the humerus, the radius, and the ulna are. And you see that even though we think of the humerus as an elongate bone, in these earliest vertebrates that emerged out onto land, their humeri were flattened broad plates rather than the long bones that we currently have now. And they're not that different in structure in reality from the lobe-fin fishes that came before them. And in this case, you have Ichthyostega, which is generally viewed as one of these transition organisms where it has a bony hind limb as opposed to an organism with both bony hind limbs and forelimbs. So when we think about the origin of the amphibians, depending on which phylogeny you're looking at, some of these hypotheses about the evolution of amphibians show a monophyletic origin of the amphibians, modern amphibians, frogs, salamanders, and caecilians, but other reconstructions have the caecilians being split off much earlier in evolutionary time, such that this is a polyphyletic origin where you have a taxon like the amphibians that has, once again, a taxonomic system that includes these descendant species, but doesn't include the common ancestor of them, which would be way, way back here. And so this is polyphyly. There are lots of things that are used in reconstructing the early phylogeny of vertebrates. One of these is the actual structure of the vertebral column. And one of the things that you see as organisms move from from aquatic environments to terrestrial environments is you see more fusion of the vertebral column as opposed to a vertebral column that is less tightly articulated. And so these are some of the early amphibious organisms. And you can see that in many cases they have structures that we commonly think of in terms of current amphibians. You see a lot of fusion of the head bones in these groups, and you see, of course, increasing fusion of the vertebral spine. So among the problems of thinking about the origin of the amphibians and knowing whether or not they're monophyletic or polyphyletic is that we don't have good estimates of the timing of many of these organisms and when these transitions actually occurred. That's largely because we don't have a very good fossil record of amphibians. The problem with fossilization in amphibians is because their bones are very thin and fragile in many cases. They don't fossilize well because they tend to get destroyed before they're fossilized. And in a lot of amphibians, the bones are not as bony as they could be. So there's a mixture of bone and cartilage, and that cartilage decomposes before fossilization can occur. And so the reason for the incomplete fossil record is due to difficulties in fossilizing amphibian bones because of their size and how they're structured in terms of mixture of calcareous portions versus more cartilaginous portions. So in spite of that, there are a bunch of synapomorphies that support putting the amphibia all together. And these are some of them. So they tend to have what we call pedicellate teeth. These are teeth that have a crown and they have these really long roots known as a pedicel, and they get replaced over time. And so you'll have one tooth developing underneath a tooth. As that tooth gets broken off, worn down, et cetera, this tooth will grow, push that tooth out. And so amphibians are constantly undergoing this process of cycling through their teeth. And that's very different than, for example, a lot of other vertebrates where you tend to grow in a set of permanent teeth. And then if you lose those teeth, there aren't teeth developing underneath them. In things like horses and other grazing organisms, you have teeth that grow throughout the life of the animal because they tend to get ground down because they're herbivores. But they're not, if they lose one of those teeth, they don't replace that tooth. It's just that the tooth that is currently there is constantly growing while the surface is being worn down. But pedicellate teeth are being replaced by new teeth coming up underneath the current teeth. This movement out of the aquatic environment into a more terrestrial environment also led to changes in the auditory system, the ability to detect sound. Sound travels very different in water than it does in air. And so you can see changes going on in the ear structure of amphibians, where you have a tympanic membrane, which is connected by ear bones, similar to the ear bones that you guys have, that then go to a vibrating membrane inside the ear that is then going to be used to detect the sound vibrations. So you have an external membrane connected to internal structures that transmit those airborne vibrations to the part of the ear that's actually going to be doing the neural detection of sound. Now, in the case of amphibians, that inner ear is connected to a skeletal structure through this opercular muscle. And that's presumably a throwback to the system that was found in fishes. And so in us, for example, our inner ear is independent of any of our skeletal structures having to do with the axial skeleton. It's actually fused to our cranium. And so that's a very different system than the system in amphibians. So I'm not going to actually expect you to know these, but just to kind of get across the idea of what kind of groups amphibians together. This idea of this, once again, the structure of the ear, this stapes going dorsolaterally from the opening that is the ear opening, where fat bodies are developing, skin with mucus or granular glands. Green rods in the eyes are not found in caecilians, but are found in all the other amphibians. Different muscles, cutaneous and bucopharyngeal oxygen uptake, so they can not only breathe through lungs when they have them, but they can also breathe through their skin, and they can also pick up oxygen from the lining of the mouth and the pharynx. And so they have a variety of ways of oxygen uptake, which also reflects their kind of amphibious sort of lifestyle. You can do cutaneous and bucopharyngeal oxygen uptake while you're underwater, whereas you can't necessarily use your lungs for uptaking oxygen when you're underwater. Tend to have short, straight ribs that just stick out kind of straight from the body. Notice tomorrow when you are messing around with your frogs in lab, feel the backbone of the frogs and then feel their ribs, and you'll see that basically in all of your frogs, for the most part, you have a rigid vertebral column, and then the ribs basically just stick out straight, such that if you looked at them from the end, they'll look like this. They're not like our ribs that curl around and meet at the bottom. They have two occipital condyles, which are the bumps on the back of your head that interconnect with your first vertebra. Differences in the radius and ulna, and also, once again, we have this general reduction and fusion of bone in amphibians. So, for example, ancient amphibians, lots of bones, they're fused together in a cranium. We still have fusion, but as we move into salamanders and frogs, you can see that the actual number of bony elements has been reduced. This is due to primarily the fusion of these bony plates that were present in the ancestors, creating skulls that were much sturdier than the skulls of the early amphibians. This fusion of bones and a reduction in the number of bony elements is once again one of these adaptations to a terrestrial lifestyle. When you are when you're foraging in water, what you oftentimes do is you forage through suction. And so what you do is you have your mouth closed, and when you see a prey item in front of you, you open your mouth. And when you open your mouth, that creates suction. The water that is outside, that presumably has the prey in it, gets sucked into the mouth, and then you close the mouth and you've captured the prey that way. And then you just generally swallow that prey whole, as opposed to terrestrial feeding, where you actually have to have ways of actually reaching out and grabbing that specimen and pulling that specimen into your mouth, which means that you need a much stronger cranium and you need a much stronger jaw structure. And you're going to get that strength from a reduction and fusion of the bony elements of the skull. And that's a general trend as you move from aquatic environments to terrestrial environments. So other characteristics that are not synapomorphies is this idea of heterochrony, changes in the timing of embryonic and juvenile development that affects sexual and mature adult phenotype. And what this sometimes results in is paedomorphosis, where you have the sexual organs of an organism developing at a faster rate than the rest of the body of the organism, such that you have the retention of larval characteristics in sexually mature adults. So if you're familiar with the axolotl, The axolotl is a type of salamander. Lives in Mexico, in springs and caves in Mexico. It is a mole salamander. It's closely related to the tiger salamanders of North America, and this is the adult form. It still has external gills. It is totally tied to water in terms of reproduction. It looks like a larval amphibian, but because it lives in these stable spring-fed environments, it doesn't ever need to transform into a terrestrial organism. And as a result, the body basically stops developing and never transitions into an adult form without external gills, but it does develop sexual maturity, and so it will reproduce in these streams. And so you have sexually mature individuals that have these larval characteristics: the tail fin, the external gills, the aquatic environment. Those are all larval characteristics. People study these. There's actually a lab down at Stowers, if I recall correctly, that studies the axolotl because of this heterochrony, this difference in developmental time of the different parts of the animal. That is what a paedomorphic organism is like. Oh, I had an axolotl in the next slide, and it's a much cuter axolotl, but this is what they look like. They do all of their breathing for the most part through cutaneous respiration and through the gills rather than lungs. Yeah, so this is part of all of that. So only bones that appear in the early development of temnospondyls appear in extant amphibians. So this is once again one of those ideas that is in biology, that the development of an organism is reflective of its evolutionary history. These guys tend to have large eyes and large eye sockets. It doesn't look like that here in this particular specimen. And once again, pedicellate teeth are part of that. These ever replacing teeth, which is once again a characteristic that you generally find in fishes, and presumably in these, you find it in extant fishes, and presumably that was the condition in the ancient amphibians, which we can't really tell that much about unless we can actually look into their jaws. So we're out of time, so we'll stop there. Any questions about this? All right, lab tomorrow starts at 10:45. I'll be there at 10:45. It's basically open. Come practice your identification. I'll be there from 10:45 until the last person leaves. Once again, there's no expectation that you be there for the whole time. Think about your own learning styles with this type of information and behave accordingly. All right.
Dr. Clint,
Yeah,
did you say the lab also be open anytime on Friday?
it can be open. You just need to let me know so I can go and unlock it for you.
Okay,
Yeah, so
sounds
if you're planning on coming, I generally won't just leave it unlocked throughout the day, but if I know that you're coming, I'll go up and unlock it.
Okay, cool. Thank you.
That goes for any of you on any days. All right, I'll see you guys tomorrow.
Dr. Kolinsky?
Yeah.
I won't be able to make it to lab tomorrow. We have to travel for basketball this weekend, so I won't be back
Okay.
until Saturday. Can I just set up an alternative time to come to lab?
Yeah, sure. Yep.
Okay, perfect. Thank you.
Sure thing.
Dr. Kolinsky, did you
Yes,
send
Ray.
my message to you?
When did you send it? Oh, is it in the chat?
Yeah, it's in the chat.
Oh, okay. You can't see anything that is on the board.
No, it's really blurry.
Oh, okay. Hopefully it's, go and look at the videos and it should be clear on the videos.
Okay.
Yeah. Are you working from home or are you working from your dorm?
No, this is my dorm room.
Ah, stink. I was getting warnings on Monday about my internet in my office being unstable as well, and Apple in my applied stats class, the AirPlay thing that I was using to protect the screens there kept fritzing out. So on Monday, I contacted Heath Hayes about both of those issues. So hopefully they're working on that. But it should be clear on the video. That's the whole reason that I do this from my office rather than from home, is because I have this big whiteboard and I can do things like what we did today, which was draw on it. So yeah, just go and check the videos and it should be clearer there.
Okay, thank you.
Sorry about that.
That's fine.
The other problem is that if you've ever run a Zoom meeting, it's
Yeah.
difficult when you're sharing your screen to see things popping up in the chat. So the best thing you can do is just unmute yourself and say, hey, dude, I can't see that.
I mean, I didn't want to, I knew it couldn't be fixed right then and there, so I just didn't
Okay.
want to take away from the time
All right.
we had.
Well, if there's anything that can be fixed during class, just let me know. And probably the best way to do that is not through chat, but just
Okay.
typing it. Yep.
Okay.
Yeah, it's one of my frustrations with Zoom is that I can't see chat very easily when I'm sharing my screen. And it's just kind of the way the platform is built.
Yeah. Well, thank you. I'll see you tomorrow.
All right, see you then.